We're going to kick off with precision subretinal delivery of gene and cell therapies by Alan Ho from Philadelphia.
Thank you. It's a real honor to be here and participate in this program. Gene therapy is happening today, and cell therapy may soon follow. We've been working on improving the consistency and the precision of delivering these therapies to target tissues. I want to acknowledge a variety of collaborator teams, gene therapy and cell therapy teams from Regenex, Gyroscope gene therapy companies, Biotime, now known as Lineage, and the Janssen Group, which evolved into Orbit Biomedical, and maybe even most importantly, the engineers from Kaleidoscope Innovation Design who helped us design some tools. These are my disclosures. The highlighted clinical trial programs are the gene and cell therapy programs that we've worked with. Gene therapy is a reality today, thanks to Albert McGuire and Jean Bennett, and we'll hear more from them in a little bit. It's very real for this patient, a patient of Nina Barakal's, Creed Pettit, because she delivered subretinal Voretigene to Creed. Creed was blind from LCA RPE65 biallelic mutation. Creed was blind, and now he can see. It's also very real for Aaron Nagel's patients. He's operated on 32 patients, delivering this to the subretinal space. But gene therapy has had some headwinds for some more common retinal conditions, for example, neovascular AMD, and those clinical trials are works in progress. The first generation trials, there were several, actually did not show efficacy, so translational scientists are working on improving viral capsids and transgene selections, and we've been working on better delivery and more precise delivery to target tissues. There are essentially three basic ways people are delivering to the subretinal space. First, the very familiar vitrectomy, retinotomy with the microcatheter and retinotomy delivering into the subretinal space. A lot of safety on that, a lot of familiarity for retinal surgeons. A new approach is a suprachoroidal to subretinal. It's a microcatheter that goes along the curvature of the sclera and a microneedle that emanates under surgeon vision control into the subretinal space. A new intriguing way of transfecting target tissues is injection therapy. Peter Campuchero and Glenn Yu and other groups are looking at injection therapies. This would be very interesting because this could take this into the office-based setting. In the Regenex next generation neovascular AMD gene therapy trial, we're enrolling patients with previously treated, high-need, frequent flyer patients. Here's a subject with 22 monthly ranibizumab injections, injecting a highly efficient and high affinity vector AAV8 that encodes for an anti-VEGF protein that is very similar to ranibizumab that's injected in the subretinal space. Here you'll see the microincision vitrectomy, one of the Regenex procedures. We elevate the hyaloid, do a core vitrectomy, and then we use this 41 gauge internal diameter microcatheter to deliver into the subretinal space. Many of you have done this kind of procedure. We do this outside the macula, about 2 disc areas away from the lesion. This is then performed, and you can see 250 microliters is not a small amount of fluid. An air fluid exchange is performed. We use the usual eye drops. No systemic steroids are given in the subretinal injection procedures. We've seen very good safety across several trials using this. One way to improve the precision and control of subretinal delivery is a new tool. It's a microdose injection kit developed in conjunction with David Brown's input. This is a 1 cc micro-calibrated syringe that is hooked up to the viscous fluid injection system so that the surgeon has foot pedal control of precise volumes of fluid into the subretinal space. To control surgeon variability, all these procedures are standardized, they're automated where available, and then we reviewed surgical video with all the surgeons to try and improve our techniques and standardize our techniques. Here you can see Jeff Heyer and Bob Avery and George Williams very excited to tell me on one Monday night video review that my idea was stupid. But we did this, and actually this was important, seriously. This was the basis for the surgical training program for other surgeons. And here are the surgeons in the REGENX trial. Similar to subretinal delivery for wet AMD, surgeons and companies are looking at atrophic AMD and delivering RP cells in suspension, or Mark Humayun's doing it on a synthetic sheet into the subretinal space. You can do it via the usual vitrectomy retinotomy. Here are 3 subjects in the BioTime study, baseline atrophy, and you can see here intraoperative photographs of the subretinal blebs of cells and also air bubbles attempting to tamponade egress and loss of the suspension into the subretinal space. The problem with these cell therapies is that they saw it in BioTime, they saw it in the Janssen trial, is that the majority of patients will develop epiretinal membranes, and in Janssen, even traction retinal detachments. And the hypothesis is that there's an egress of the cells that then creates the membranes, and that egress occurs through the retinotomy. That failure stimulated multiple approaches and a lot of the Janssen surgeons to develop new approaches to the subretinal space. This is an animation of a suprachoroidal microcatheter. You can see here the catheter and a flexible microneedle that then emanates into the subretinal space to create the detachment and deliver cells. The advantages of this is that there's no vitrectomy needed, there's no retinotomy, and therefore you might get more precise and consistent dosing for these patients. Here you can see the innovation kaleidoscope design of the hardware, very fine. Here's a video showing this procedure. I've shown this and it's evolved over the course of several years. Let's see. You do a microincision and chandelier, no vitrectomy. A sclerotomy is performed with a cut down 6 millimeters posterior to the surgical limbus. The microcatheter is then fed underneath vicryl loops to stabilize the path of the microcatheter and to reduce the degrees of freedom of the internal catheter so that there's no damage. You do this outside the eye, switch to the microscope, and then view the microcatheter passing posteriorly. The microneedle emanates, like you see here in animation. A saline bleb confirms that you're in the right subretinal space, and then you switch the lines to the cell therapy. And now you deliver the cells. Just a quick animation showing the engineering firepower from Kaleidoscope. And now this is now approved, FDA approved, and being used in the BioTime trial. Two-dimensional imaging is very important here. Here are the Janssen patients. This is really important for atrophic AMD. And three-dimensional imaging is going to help us get into the right space precisely and with safety. You can see here the microcatheter in the subretinal space, delivering cells safely and in the subretinal space. You can then take this and measure dosing, which is an important metric in a clinical trial. So to summarize, gene and cell therapy is happening. Subretinal delivery via a vitrectomy and retinotomy is very familiar, but you can have variable dosing with efflux, and particularly in cell therapy trials. The suprachoroidal to subretinal approach has been approved by the FDA and is being used in a clinical trial. Imaging will be important, and we have 3D imaging for dosing. Thank you.